Hello, everyone. Welcome to BrainMap. It's my great pleasure to introduce our speaker today. Our speaker really needs no introduction. You probably know him better than me, but our speaker is Professor Xin Yu. So Xin Yu studied neuroscience and biophysics at New York University. After obtaining his PhD in 2007, he moved to Bethesda and worked at the National Institutes of Neurological Disorders and Stroke, NINDS. He combined memory and bold fMRI with electrophysiological recordings to study the system-to-synaptic brain plasticity. Using high-field fMRI, 11.7 Tesla scanner, he made a significant effort to push the spatiotemporal limit of fMRI so as to better understand the neurovascular coupling mechanism of fMRI. In 2014, he moved to Germany and built the Translational Neuroimaging and Neural Controls Research Group at the Max Planck Institute for Biological Cybernetics. The group specialized in developing advanced fMRI methods under high field, 14 Tesla for animals and 9.4 Tesla for humans, and implementing optogenetics and fiber optic mediated calcium recording methods to build up multimodal fMRI platform to study different brain states, arousal, anesthetized, and sleep. His group also focuses on investigating the neurovascular dynamic changes underlying the coma induction and reemergence, so coma induction and reemergence in rodent models. In 2018, Dr. Yu moved to Martinos Center and continued to develop the multimodal fMRI methods for both basic and translational studies. As always, BrainMap Seminar Series is co-sponsored by the P41 Funded Center for Mesoscale Mapping housed in the Martinos Center. With that, I'll pass it over to you, Xin. Thank you so much for being here with us today.
Sure. Thanks about introduction. And next time I'll bring a shorter version of my bio. Seems like you read through all my bios. And thanks, that's perfect. And okay, so since many of you may already heard something about this, so I will just directly get into what I want to talk about today. All right. So does it work? Okay. So you can see, right? So today I'm trying to present something really new. But before I present it, I really want to go back to give you a little bit of history, at least from my trajectories about why I'm becoming interested in this topic. So this is a work I done during my PhD. And we always, since I started my research, I always dreamed if there's a way for me to map the brain functions, right? Directly, indirectly, right? So at that time, the first things I get to know is the manganese-enhanced MRI. Many of you even work on it, right? So I don't need to give more introductions. So the one effort I made is that I look at the auditory systems, right? So seeing how the different frequency, the tonotopic activation can activate in the brain using manganese to mapping the inferior colliculus. So at that time, it's very exciting, right? Consider this is exactly 20 years ago when I published my first author paper, basically. So, but there is issues there, right? Because manganese cannot passing through the blood-brain barrier, and typically, even though the way it definitely work, but it takes a lot of accumulation times, right? Like we spending like overnight, right? Continuing activating the pathways, allowing certain kind of accumulation, which is more like 2D glucose, 2D Gs started, right? So it's not really translatable, and definitely, gradually lost, the tractions and indicated from the citations here. So, but if, like I mentioned about, if this is a dream, right? I definitely keep this dream for the past, academic dreams for the past 25 years. So I just briefly give you a little bit, what my journey, right, is particularly in the preclinical high field, partly covers the ultra high field human studies when I was in Germany. So here, what you can see here is I just showed you MEMRI, right, can be used in a certain way to map the brain functions, but everybody knows, right, here's Martinos, right, so we have a bold, we have fMRI developed here, right, so which is really give the way, right, to map the brain functions, but we also know the indirect coupling mechanisms, which certainly give us kind of barriers for this method to be fully implemented in the clinical practice. Even nowadays, right, there still be challenges. I think I will show you some kind of insights in the past 2 decades, how we are presenting the underlying mechanisms with the ultra high field MRI. And like I'm showing you here, right? So what I'm really excited is about this part, right? So I'm going to spending significant amount of time to introduce you what we are doing. Okay, so let me really get into the results. So I will spend at least 10 to 15 minutes, right? Really not about the topic I'm going to show you, but I think it's very crucial for you to understand it. Why is us, right? Why is us, it's me, or at Martinos, right? We are making these things happening, okay? So first, let me show you, how we are working on the proton, based in the hemodynamic fMRI. So this is some studies I did it also a long time ago when working with Alan Kresge at NDS. And you can see, right, this is a coronal slice of the barrel cortex. We give a stimulation of the whisker, okay? And then you can see this is actually a very high 150 micron in plane every 0.2 seconds sampling rate. Right, whenever you give a stimulation of the whisker, it is the barrel cortex, right? So this is the barrel cortical region. So let me go back, right? So you can see, there's very strong, this is the bold fMRI signals, beautifully presented, right? When I first think, I was so excited because I was trained as a neuroscientist at barrel cortex, right? I told Alan, hey, I see barrel, right? This is the barrel, right? Barrel cortex. And he laughed at me, right? So, hey, that's not barrel, right? So if you look at the raw data, right? This is upper raw data, you see the black stripes. What is that? So actually that is a vein, because the T2 star, right? EPI, this is EPI sequence, right? So, you see the T2 star waiting, right? The vein has a lot of deoxygen glucose, deoxygen hemoglobin, and which really give you this fast T2 star decay, make it darker, okay? So if you see it, right, this is just very nicely matched, on this penetrating veins, right? It's not surprising, of course, right? This is a bold, it's coming from the deoxygen hemoglobin changes. But, at that time, right? So I was really interested, hey, what it turned out, I learned the lecture, right? We know bold is coming from hemodynamics. This is the first time I really get into, it turned out we are looking at vessels. Optical imaging study has been doing this for years and years, right? So, but there's some tweak there, right? So one thing what I want to show you is, then I alter the slices. I just want to see more, right? And then all the stripes become an individual dot, right? These T2 star weighted dots, okay? And I use a very old method. Afonso, used to work with Alan, and they published 2002 paper. Many of you maybe know there's a science paper published. They pretty much using the exact same methods, right? They claiming they see the activities. We are doing the similar way, but in very different scenarios, right? And let me quickly show you what exactly we're doing, right? Consider it's a case space, right? Typical EPI, you get a few seconds or hundreds of milliseconds, but now we only acquire single case space time. Then we move to the next one, during this block design, on-off design, right? So that we can make it very, very fast, a few milliseconds, no problems, as you know, okay? Then, if we keep the stability of the mouse physiology, everything is anesthetized at this point, okay? We are able to fill in the second case space line, multiple case space lines, and then we can reconstruct the images. So that's why I call it reshuffled there's KT space acquisition, right? By doing that, that can gain us a very high spatial and temporal resolution. I'm going to show you the bold fMRI, what we are capable of doing. But before that, right, because like I mentioned, I'm quite interested in the vascular origins of the bold signal, right? So I ask myself, how can you really differentiating the arterioles and venules, right? So then, we implemented another method. Everybody know multi-gradient echo, right? I'm not in details, right? But the point here is by alter the parameters of the TRT and flip angle, we're able to differentiating the arterioles as brighter dots and venules as darker dots based on the inflow and the T2 star weighting features. Okay, so now let's use the reshuffled KT space sequence, right? And this is the image, covered full power, right? When you give a stimulation, so this is the 100 micron in plane, 500 micron thickness, 0.1 second TR. So at that time, right, you can see we published over 10 years ago. It's very, very fast. So what is really interesting is you are seeing these bold signals beautifully overlapped on these penetrating dark dots, which is penetrating venules, exactly followed what I just mentioned with your bold oxygen changes in the vessels, the venules, which is the most significant. Similarly, you can see the CBV right here, in Martinos, right? So that's the CBV signals. We inject iron particles, not the GAT, like originally, Benevilles did, right? But similarly, right, you can see the vessel dilation now, right? And where they're coming from is arterioles, because the smooth muscle cells, they're dilating their iron particles into the brain. They're inducing the more field distortion in the local voxels, right, with arteries there, right? That's why you see these blue signals. It's weighted. I just want to show the large field of view. This is the advantage of fMRI, right? Because you can see much bigger brains, much more penetrations, right? You can really see these both signals. I'm pretty sure you see that, right? So from this white box, you have a 7 times 7. This is oversampled, right? But you see this 10 Hertz sampling of the both signals, right? From this individual penetrating venues. And you can even see some drilling veins. You see that? You see this? These drilling veins, some darker, some blue signal is also meaningful. My coworker Yuan here, right? So he also published a paper talk about what happened, why you see the blue signals, which is negative bold, right? Very nice paper. So not only that, you can see CBV, right? So like I mentioned about this is CBV weighted image, right? That's why kilostar signal is decreased, right? You can also see how exactly the individual, CBV signal looks like. Okay, so I want to give you more insights, right? When you have a resolution high, right? Then how the both CBV looks like, right? Not only from the evoked perspectives, but also in the resting state perspectives, right? So, everybody knowing resting state here, right? Very simple way, state-based, right? You have a voxel there, you're correlating with all the other voxels. If they are functional connected, they are going to be show stronger correlation coefficient, right? But now let me show you how it looks like from the single vessel perspective. Whenever my cursor is, that's the seed voxel. You see, then when the cursor on the venues, all the other venues is heavily correlated. You are going to see that, right? So actually, what we are really showing here is I'm not talking about functional connectivity. I'm talking about venue network connectivity, right? Because your resolution is high enough. Not only from the single vessel bolt, how about single vessel CBV? You see that, right? Whenever your seed is located, at the arterioles, which is white dots, all the other vessel dots is connected, right? It's heavily correlated, which is arterial network connectivity, right? So, but of course, I'm not denying there is underlying neurovascular coupling features. What I'm trying to say is we are able to see inside to really get into the vascular origins with the conventional hemodynamic fMRI. And that also will be contract to what I'm going to show you with sodium, right? Just bear with me, right? So here, we have the time series assay, right? We can see the frequency, which is anesthetized mouse. That's why the frequency is very, very low, right? High frequency is nothing there. We can do it in human. This is really nothing complicated, right? So here is a 3T, right? We get it in 800 micron implant, 1.5 millimeter thickness, right? You can see exactly the same thing like what I just showed you in the mouse brains. However, the seed is based on the sulcus vein, not inside the penetrating vessels. Our colleague John Polymeti, right? They are doing this, using 70 right here. Now he's in Stanford, right? So they are pretty much reproducing this data in human brains with the 70. So, but at that time, right, we are really showing a beautiful, this so-called venue connectivities. We can even do assay reconstructing these heavily correlated venues there, and we did actually performing using 9.4 with a 500 micron implant, 800 micron thickness, we are able to identify, you see the seed-based, the intracortical connectivities, from the other penetrating, arteries, venues, in the human brains, right? So you can see this is I think it's 200 micron implant resolution, anatomical data. You see the penetrating venues there. Okay. So everything what I just quickly showed to you is what we did back in Germany, even back in NIH, right? So here, right? So we have really take advantage of our 14T, right? Thanks for Jerry, right? Really providing this very good platforms, allowing us to really grab stuff so quickly, right? The first things I want to show is we really build up a weak mouse platforms. I think I don't even need to say too much, right? You can already see that, So this is led by David Haik and Xiao Chen. And so you can see, this is highly implanted coils is very important, right, in the future, right? Why we can gain the SMR? Because we really try to reducing the potential artifacts and try to really have the coil secured on the mouse head to producing a really high resolution. Here is a weak mouse, right? So each EPI image, right, we can acquire 100, 200 micron resolution. Left is pretty much the same resolution as acquired anatomic image. So basically, we can get the anatomical resolution level fMRI data, right, from a weak mouse. So why I think it's striking, exciting is because 20 years ago, 25 years ago, when I was a PhD doing the mechanism-enhanced MRI, right? It took me 2 hours to acquire the same spatial resolution image, right, when I was a student. But now it took 2 seconds for David to get pretty much similar resolution in a weak mouse. That's how we have revolutionizing the preclinical side, how the fMRI can be approved, okay, can be achieved. So not only that, we can even inject iron particles into the brain, right? We can achieve 100 micron isotropic data sets, for anesthesia mouse brain, right? I just want to show you, see the left, you can see the penetrating wave. This is CBV. We will activating the left forepaw, or we will activating the right forepaw, right? You can really see, the vessel specific. But now it's not a single size, it's 3-dimensional, 100 micron isotropic. So we are approaching from hemodynamics, right, from proton-based, very high levels of specificities. And we can also, doing the resting state analysis. This is averaged, right? This is resting state. We can see that in specific components, right? You see the SA, right, which is nicely coupled on these individual vessels, and we can see the multiple frequencies appears, right, from this particular vessel dynamic changes. Okay, and this work really gained us, have a great collaboration with David Kleinfeld, and so the reason I'm talking about it is because, so I think this is, from my view, this is real very good directions from the future fMRI studies, right? So this called singular value decomposition analysis, which really deal with the spectral specific analysis, right? Which has been very much used in the neuroscience field. But now I think by working with David, right, we are able to really see how the spectral spatial oscillation features appears in the brain with fMRI, but this is based on hemodynamics. This is my last slide. I know you come here, listen to me talk about sodium. The only reason I talk about here is, based on all the efforts we have been made, right, improving the resolution, improving, how we can see the vessels, right. I'm going to show you 2 studies right now, right. From my view, right, we are pushing forward. One is we are able to using the so-called SVD to see how the visual dynamic propagation into the brain, not just simple resting state connectivity changes. We see the spectral specific propagations. Okay, consider its current in the ocean, right? How exactly, right, they are moving, right, in a living, a weak mouse, right? Not only that, we can also see how 2 animals, when they were interacting with each other, that's the bottom part, right, where I'm going to show you, right? So we are simultaneously imaging 2 weak mouse where they are interacting with each other. And we extract very specific center frequencies at 0.7 hertz, and it appears they have a phase offset between 2 animals, right, in this prefrontal or simulated cortical regions, right? So these 2 part of work really highlighting, from my view, from preclinical side, how potentially we can using these fMRI studies to go deep, to really reveal something other technology cannot achieve. However, there are issues. Like I said, when we talk about all of these dynamic changes, right? So I didn't get into, forgive me not getting into the inside, the details, but always the issue is how can we decouple the so-called vascular dynamics, the visual motion, for example, from the real neuronal coupling features? It's always interleaved. When you see the neural activity, you will puzzle the whole. Are they coming from the vascular spreading issues? Or when you talk about the visual dynamics, you will say, oh, they may be coupled with the neural activities, maybe regulated by all of the non-adrenergic or the atomic, regulations features. And similarly in the lower end, right? When I tell you that, hey, we see some reciprocal activities, right? You may say, hey, you have fMRI signals. Are you telling me you can see the 0.7 hertz near 1 second responses between 2 animals? This is always the questions there, right? Really, make us to think something new. Okay, sorry, right? I'm spending more than 20 minutes to say something I'm not even supposed to say, right? I mean, you're expecting to say this, but I think it's very, very important. I give you this foundation, not only from the mechanisms perspectives, but also from technical perspectives, why we want to do this, okay? I don't need to say more. So this sentence I'm writing here is really not a statement rather than a hypothesis or speculation, right? So is there a way for us not to use protein instead of using sodium, because we know sodium is so much important to get into the action potential inductions. And we also know that, whenever the action potential inductions in the mouse, in the brain, right? So there are a lot of movement. Is there any way, right, we can detect some signal changes? Now, I'm only talking about the single quantum signal changes, because instead of the multi-quantum, this number one is very even worse SNR, maybe less than 1/10 of it. And, it's much harder for us. So this is something I'm going to share with you, how we see it, okay? But nevertheless, we need to really admit it, right? This is not like something new. That's what I'm saying. This may be a century old, right? If not century, at least 30, 40 years old question. Maybe many people are trying it, but it's just hard. Why is it so hard? I just lay out some very basic facts. So we know that we can imagine brain nicely with the proton because it's just high concentration of the proton in the water, right? So, but sodium, even though it's the second most abundant NMR nucleus, but compared to concentration in the body, right? So we have a mean value, right? I'm talking about extracellular, intracellular sodium, right? Around 40 millimolar versus over 100 molar of the protons. So that's already a big ratio difference. Not only talk about their NMR sensitivity differences. So altogether, consider we have 80% water in our body, right? So we are talking about over 20 to 30,000 ratio SNR difference. That's a problem. So how can we beat with this number? It's almost, you need 100 Tesla. Maybe it's still not even enough if you purely think about this 30,000 ratio difference. However, I need to share with you more to really shrink that ratio differences. First, let me give you some of our own measurements, right? So this is the first, I believe, our real T1, T2 measurements of sodium 23, right? In a living mouse brain of the 14T, right? So basically, the take-home message is, we measured in the CSF or measured in the cortex. CSF has more sodium. It's more like extracellular, right? Near over 100 millimolar versus the cortex, right? So you can see their typical T1 is lower than 50 milliseconds. Very important. And we know sodium 23, right, is 3 half spins, right? So there's a quadrupolar decay there. So there's a T2 short, T2 long. I'm talking about the single quantum. Okay. So we measure them, which the T2 short is less than 2 milliseconds. The T2 long is, varied from 10 to 30 milliseconds. Okay. So just bear this in your mind. So now let's make these comparisons why we can shrink these, very formidable ratios there. First, let's talk about the hemodynamics. Typically, doesn't matter in animal, human, right? 30 seconds epoch. Give stimulation for 30 seconds. Very makes sense, right? So that's like one epoch. But now, if we look at sodium, if we want to see some real neural activity changes, we can make it 300 milliseconds. That's already over 100 times differences. I'm talking averaging, of course, and the square root. Okay, so that's what I'm trying to say, 100 over 1, but we are square root of the 100, okay? Second thing is, hey, we need to think about it, right? How you define a TRT, right? So if you, typical proton, right, 4 seconds, right? But sodium, like I mentioned with you, 50 milliseconds, that means what? You have very short TR, right? I'm talking about 10 milliseconds, 20 milliseconds, right? The sodium can easily relaxed in a decent amount of, their longitudinal momentums, correct? So that's also another advantage. But you need to think about T2, right? If you say, hey, come on, right? The T2 of proton is much longer than the sodium. But remember, I showed you the CBV. What did I do? I inject iron particles into the blood. I show you the beautiful response there. And their T2, I don't know anybody ever measured carefully in the blood, right? But in my experience, it's less than 2 milliseconds, at least in the ultra high field. So that's also very much to sodiums what we are talking about it. Okay, so the real player is the spatial resolution, right? If I tell you, I think I proved to you, hopefully in the past 20 minutes, right? We can get near 100 micron isotopic CBV fMRI. We just simply increasing the increasing the voxel size, right? So now, if we're just using the 600, 1.2 micron, right? we can even easily get a 200 times ratio SNR increase. And not even talking about our RF coils, right? We make a better RF coils, really have to figure it. I'm going to show it to you why we use that, right? So that also can gain us 2.5 gain of ratio, right? Altogether, we can shrink these very formidable ratios up to single digit. I think that's also, fairly speaking, right, there shouldn't be any argument, right, because this is a very simple way to really imagine that. So then let's coming back, right? So previously I mentioned about it, right? We can do the CBV, right? Consider one trial, right? It takes us for 7 or 10 minutes, right? If we know there's a ratio of 1 or 5, or even, consider by calculating how many averages, we can really acquire comparable SNR, not CMR, okay? Because I don't know what CMR is. So we can get 6 hours to make it comparable with the 7 minutes of photon. For human, we don't know, right? But it's not like it's totally, unpredictable, right? Because we have voxel size gain to play, even other typologies to play. But for animals, it's feasible. 6 hours experiments. In my past 25 years, I think that's the lower end of the scanner time for me, right? Of course, here we have to pay scanner fees. That's a different story. But most of the time, we can find a way to, thanks for our administratives, right? Find a way to get compensated, right? So it's definitely very much feasible here, okay? So first, I want to show you is the proton anatomical images, right? So that's also from Nguyen's work, right? So what you can see here is, it's very simple, right? We got MCAO mouse, right? So now, we can just get in. This takes, I don't remember, 80 minutes or 2 hours. I don't remember. But you see the resolution near 150 micron, 500, right? We can also get a 300 micron isotropic resolution there. You just need to know this, right? So now we review the literature, right? So there are 21 tested image of the rodents, right? Comparable study, but their resolution is 1 millimeter isotropic. Okay, so they have a 30, of course, they have a 30, 9 minutes, 30 minutes, right? It's a bit variable, right? But what really I want to say here is now we already make the sodium preclinical image feasible, right? Approachable, very comparable spatial resolutions, which have never been achieved. Okay, so now let's come back to the real story, what I want to share with you today. Remember, I talked about the reshuffled KT space, right? So this is the key, okay? Why I'm saying this is the key, right? Because we need to really get a very fast image, right? So now even whole brain, cover majority of the brain, right? So how we did it is we set the TR at 10 milliseconds, we get 1K space, we move to the next image during this block design, right? If we have a 300 milliseconds block, right? Or epoch, I call it, right? For example, I'm waiting 50 milliseconds to give a stimulation. This is the 4-pole electrical stimulation, right? Typically, it's like 2 milliamp, right? 300 microseconds of the biphasic response, electrical shock, right? So that's very typical for rodent fMRI, right? And then we can give a left or right, we can alter that like event related, right? And then by repeating once, twice, and many times, right? Depending on the KY and the KZ numbers, right? So that's how we can get the one set of image. But because the TR is too short, so typically, if we can get 600 micron, 900 micron, 1.2 millimeter thickness, right, cover the whole brain, right, it takes us 1 minute, okay? But that's one trial. But you can imagine, right, there's really no signal there. So you're repeating this one trial for hundreds of times, at least 64 times. So consider that each trial, you have to give it intervals, right? You don't want to repeatedly stimulating the mouse, right? Then that's why I'm telling you, we need at least 3, 4, 5, 6, 8 hours. Okay, so that's how, how we get the data. So this is the first, for me, is the most striking results. I even presented it, right? In the ENC, Matt Rosen was chair of the, the ENC conference. And what you can see here is the lower end, right? Remember, right? I give a left whisker stimulation or right 4-pole, right? Ba, 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 right? So now I can do event-related correlation, right? First, I show the lower one is a bold signal, right? Same animals, okay? So the activity there, and then I see, I'm going to tell you how I get a functional image, just show the result first. I see some kind of correlation, negative correlation, very interesting at that time, near the barrel cortex. And the opposite, I can see the forepaw, right, region in opposite side. I also see some negative signal there. At that time, it's really amazed me. But you may ask, how did you get the correlation maps? Because my assumption is, if I have a 300 milliseconds, right, there are any responses, right, it will match very close to log field potential data. So because if it's action potential happening there, right, it may occur in the first few tens of milliseconds. So basically, I use the local field potential responses as my ideal functions. And then I do the correlation. That's how the correlation map looks like. Okay, but I can assure you that, right, you can see, right, that has been done a few years ago, right? And, when I look at it, this is a bit circulating, right? But based on the ROI, right, when I look at it, right, I do see there is definitely this sodium image, sodium raw image, right? There are signal drop, right? Either from the ROI, this is from the ROI, not from the active areas, okay? So, or from the left forepaw. So this data, right, really struck me because at that time, I don't even know why, right? Because I didn't have any speculation. It should be positive or negative, right? I feel like I want to try it. So, but of course, right, I tried reading some papers, right? One paper published very early, over 15 years ago, right, by Itman. And but they are really showing, hey, sodium, maybe look at CBF, right? But really not talking anything about neuroactivity. There are some human studies in France, right, just a few years ago, right, they also try to use the reshuffled KT space UT sequence, but not really giving this very, very fast, measurements there, right? So, but since we look at it, right, so, we really want to see, is this reproducible, okay? So let me show you this. So, but there are some other stories, but I will hold on this, right? So this is according early this year, okay? So basically, this is really raw data. Individual animals, you can see, this is rats, 4-week-old rats, had coil implanted, okay? So, so both signals, I have set resolution similar to the sodium, okay? 600, 600, 1.2, right? You can see if I activate the left forepaw, you see the both signal there. And correspondingly, you see some kind of negative correlations, in the NARS, right? The other animals, you also see something there. The other animals, you can see there is kind of very comparable, negative signals detected in the corresponding active regions. So the lower end just see the horizontal view, right? This coronal view, horizontal view, right? You are seeing, some of the signals really nicely, quite correlated, with what we want to see. This is individual animals results. If I look at the time courses, right, you are seeing that when you give a stimulation, right, first 20 to 30 milliseconds, you start seeing this signal drop. This like 1%, 2% signal drop is as what we observed. And also we tried in mouse. Okay, we just don't want to say, hey, this whatever reason, right? We really, I talked with Bruce, right? He wasn't, hey, you want to make sure this is not like something just random, right? We tried many different scenarios. I'm going to tell you that later, right? So this is a mouse brain, okay? So you can see this is a raw anatomical 200 micron plane, right? See this multiple slices. When you smooth it in the matched spatial resolution as a sodium, you can also see where the both signals are. You can see there's negative correlations in the active regions in the sodium, okay? And if you opposite, the different animals have opposite side, right? Activated, you can also see, right, the both signal there, you can see some negative sodium signals, okay? So if you look at the time series, right, you can see 20, 30 milliseconds, you get a signal drop. So I want to show you this older data we did it, right? This is the rat data, okay? So when we give, this is exactly the same stimulation of the forepaw anesthetized, but this anesthesia, by the way, everything that shows anesthesia, anesthetized, right? We use these dexmedetomidine IP injections with 0.5 to 1% isoflurane, right? So there's a bit of leaking, right? So, but typically, right, the mouse is lightly, anesthetized. I'm going to show the respiration rate data. It's very crucial. Okay, so what I'm trying to share this with you is very comparably when I do log field potential, right? Near the 20, 30 milliseconds, we do see the typical log field potential deflections there. But I want to say something. What I believe is not because of potential changes causing the sodium, it's because the action potential changes cause the sodium. Okay, so the reason we have a time very much aligned is just because during the log potential changes, right, the more neurons firing, the more action potentials occurred in a population of neurons. Okay, so this is my understanding, right? But of course, we can discuss more about it. Okay, now I didn't show the raw data, right? I need to show you the raw data, right? So this is the average, raw data there, right? Like this is because we have a 300 milliseconds window, right? And now this is 300 milliseconds. Okay, so we have like 30 time point. This is the movie corresponding whenever you can see how the movie runs there, right? So there are some tricks I want you to know, because if you see the timeline, right, there's actually over 1 year, right, we couldn't get a good results. Okay, after I see my first exciting data I share with you left and right. I really puzzled at that time, right? I don't know why until this happens. Then almost every time I see it, which is how to deal with noise, right? So we all know, right? So we are doing imaging, we're not doing optical imaging, right? The respiration, right? They can alter the B0 field, right? We call the respiration-induced B0 offset, right? Even though your RF coil is somewhere here, right? Your chest breathing, right? They can alter the field, which can interfere far away from your, from your detection areas. Okay, but that one you have to do proton, right? It's not a big deal because SR is high, right? But now we are doing sodium, right? That combination is definitely killed the very small amount of signals can really achieve. So everything what I showed you here is I do a respiration gating. That's the key. So that's why you may see here, the first time point is very high. Why it happened like that? Because each 300 milliseconds, right? When you do another one, the first couple of points, they are not experiencing the saturation effects anymore. So they relaxed fully, right? So that's why you see the field is very high there. That's also why we have to change in where we give a stimulation. Okay, but I can assure you, right? Everything that I showed you, we give it a 100 milliseconds, 50 milliseconds, 150 milliseconds, 200 milliseconds, right? We varied it. We were always seeing the corresponding responses. That's further confirmed what we are doing. But not only that, I also want to show you everything that I mentioned, it makes sense, right? Because if everybody read this science paper I published a few years ago, now they got retracted, right? I shouldn't call it a science paper anymore. However, the same author publishing another paper, which is, I think, is very well drafted, right? If you get a chance, you should read it, right? So they're talking about why they think they see something which is not reproducible. They trying to using the so-called RF splitting issues, okay? They even do a simulation, trying to understand it, right? If you have very short TR, right, multiple repetitions, right, how potentially, right, if any kind of RF spreading related problems involved, right, you may get some unwanted echoes or whatever reasons there, right? So that is something I think is very well thought. That's also originally why it got people excited. Okay, so now I want to tell you is when we do the experiments, right, because we cannot control the respiration rate. We have a 300 milliseconds, right? So this is a real time to monitor each respiration rate R, okay? So you can see here, right? So this means the duration, right? First and second, the mouse, you already have 2 hertz, right? 2 to 3 hertz, right? It's latency sensitized, right? That's why you see they always have like 400 or 500 milliseconds. And that signal is always changing. What does this mean? This means each 300 milliseconds, we have a varied intervals. It's never be same. So that will what? That will cancel out all the potential accumulated unwanted echoes or whatever the RF splitting related issues there. So we don't have that problems. Okay, so this is very, very crucial, right? Just want to give the ideas, right? How we are able to control our conditions and producing something which is meaningful. Okay, come back, right? I showed you the data, right? Some of the animals, who knows? So now actually we do a group analysis like human studies, right? So here you can see we have 9 animals, right? This is the rats, 4-week-old rats. I'm going to tell you why we use 4-week-old rats later, right? And we have time. So we can see the average, both signal there. We can see these corresponding, sodium signals, right? Which is group analysis. We have the mouse data there, right? You can see how the both signals appears and, nicely highly reproduced from the different group from the group analysis. Okay, now if I can stop here shortly to tell you, this is more like, hey, our exploration to see, we see something meaningful. But we certainly need to validate it, right? Is this something real or something just unwanted issues? Allow me to introduce you to another effort in our lab, right? We're doing the multimodalities, right? I use 2 or 3 slides to introduce you what we're doing. So some older studies we did is, we have this genetic encoded fluorescent calcium sensors, for calcium, even for glutamate, okay? So the older studies I'm showing you here is, this resting state anesthetized, right? You have this bursting from the potential data, right? You can pretty much see 1-to-1 match, right? From the neuronal calcium transients. Okay, so this is something many people did it, right? So now it's not good news, it's not big news, right? So not only that, you can also give a stimulation, right? So here I'm showing you is left and right, right? We have the GCAMP calcium sensor, we have glutamate sensor. You can also measure glutamate. So you can see the evoked both signal there, right? You can see the evoked calcium transients or the evoked glutamate spikes, okay? So, and when we look at the details of the responses, right? Compare the neural calcium or the glutamate release, right? You can see the glutamate has much sharper, for always have maximal, right? So that's more mimic to the loss of potential data. This is also not something new, right? People already did it. But just in our lab, we're doing fiber photometry all the time, okay? So why I'm showing that? Because that can be a perfect way for us to perform them with fMRI, right? I already showed you we are, right? This is another example, right? From here, we acquired it from Martinos, right? So the previous data is from Germany. So you can see here, right? We can see a beautiful both signals, optical fiber there. See that, right? You can also do local potential data there, right? And you can see this 1-to-1 very nicely matched glutamate signals. So all of this allow us to simultaneously recording the evoked glutamate signals and the sodium fMRI signals. But if you remember, I told you before, right? Each trial is near 1 second, right? 1 minute, right? You have a respiration gating that can be even elongated. I'm showing you here is trial 1. This is evoked glutamate signals. This is enlarged. You can see how the individual glutamate signals looks like every 200 milliseconds. And then we're repeating them for hours, right? We can try 100 or 100 trials. I just want to show you, we always seeing this evoked glutamate signals indicating there are evoked neural activity, right? Because it's hard for us to look for potential with fMRI in our current setup without any kind of confounding by the gradients, okay? So you can see, right? Obviously, you are giving this for 2 or 3 hours. There's desensitization, right? You can even see the evoked signals get reduced. So that's perfect. I don't even need to change the stimulation levels. I can use the amplitude to plot the glutamate signals versus the sodium signals. And then, oh, by the way, right? So this is individual animals, right? You can see the correlation from the sodium. This is the bold fMRI. You may wonder what's going on. There's very bright areas. It's because of fiber insertion, right? It's lesion. Sodium is always good. Remember I showed it to you? If you have any kind of edema, cellular edema, right, sodium is going to show very bright. So this is not like something, that's why there's a human studies always think sodium can be a good way to look at it, injuries. Okay, so but SNR is low, right? That's what we're fighting with. So, and this is something I want to share with you, right? So now if we're grouping this, glutamate signals, right, based on their evoked magnitude, and then we compared with the sodium evoked responses, right? We are actually seeing the stronger the glutamate signals, you see more negative of the sodium signals, which exactly matched what I told you with the negative responses, right? So not only from individual animals, we tried 6 different animals, we are seeing this very nicely, reproduced results there, right? You may wonder what's going on from your x-axis, right? Because typically, right, if you see sodium peak, right? See here, I'm showing 2%. And now I'm doing the z-score. The reason is, it's not always we are lucky at very good responses, right? Some of them are very small because we have to alter the baseline levels. So some of them make 0.03%, some of them, 1.2%. It's very difficult for us to show it. That's why we do a z-score, okay? So there's always a reason there why we're doing this. So not only that, I also did something is when we don't give any stimulation, right? Just every minute, right? We just acquiring that, right? starting from the field experiments, actually. But we're seeing this glutamate signal, right, have a beautiful oscillations. This is the raw glutamate signal looks like, which very much makes sense because if you see the anesthetized mouse brain, right, you do see low potential, right, the brain state is always changing. So that always showing this kind of oscillatory patterns there, right? If you look at the oscillation patterns, not always the same, right? We can call it desynchronization or synchronization, right, of the, of the brain state, right? But what does that mean? My understanding is sodium in and out, right? They have a more like a wave-like features. So higher frequency means maybe more sodium movement. I'm purely speculative here, right? Maybe more sodium movement. Low frequency means at a given time, less sodium movement. If that's the cases, we can easily plot, the center frequencies of the oscillation as a function of the mean sodium signals in the cortex, in the mouse brain, right? And we also see there's actually some negative correlations there, which further, strengthened, my confidence. Hey, whenever we see there are decreased signal there, right? Maybe it's because of sodium movement. Maybe it's relevant to the neural activities there. Okay. So last few slides, right? I want to really talk about what truly happened, right? From the mechanistic level, right? How can we get insights based on the preclinical data? Hopefully, we can drive it in the human studies, right? So we all know, hey, because I repeatedly claiming this action potential, right? Then one simple thing is what happened during action potential? Sodium influx. Are we talking about something changing intracellularly from the sodium ions? Maybe not. Why? If you do ChatGPT, right? So now it's easier. Otherwise, you have to look literatures, right? So there's a very good, nice equations educate you how you can calculating per action potential, how much sodium will get into the cell, which is less than 0.07 or 0.08%. That's like over 1/1000 of the intracellular sodium. What does that mean? If we have a cube, which is a 10 micron cube, right? Then basically the sodium influx only take 1 micron in your cube, right? That's very small amount of sodium, and if purely from the concentration perspective, I don't think that's really a good way, maybe contributing to it, right? Because you get in there, maybe the overall is still the same, but they maybe experience more altered environment, they have faster, shorter T2 cell decay. But I think that effect will be very, very small. Okay. So, you can also talk about, is there a kind of osmotic swelling there? That's also very hard because typically, right, you have repetitive stimulations, right? I'm talking about seconds, right? You may see some swelling, but per action potential, right, there's also ChatGPT results, right? The volume change is also negligible. It's even less than 1/1000. So, but what else, right? So I'm not telling you that I have the full revealed everything. That's hopefully will gain us more R1 grants to really investigating them, right? But I think it's made relevant to the diffusion, right? To how exactly the movement, right? How the sodiums interact with the microenvironment. Okay. So I give you a very naive way, right? I'm not trying to, I know from the MR experts' eyes, this is too naive. But if you see the paper, right, by Bill and Charlie, 1991, right? There are two very good papers, right? They try describing the multi-spin, multi-quantum perspective, right? But I want to use a the simplest way, what I can understand it, right? So this is a typical FID, the quadrupolar single quantum FID, right? So typical T2, right? You have one term, now you have two terms, okay? T2 long and T2 short. Meanwhile, you have also the partition coefficient, right? Which is relevant in how you can estimating the FID. So we are able to, by collaborating with Anna and Martin, right, from BU, right, when we have an organoid, right, there's no blood, there's no motion, right? And then we are able to really start to measure how exactly from single quantum perspectives, right? The typical T2 short, T2 long in the organoid, when we are able to controlling this cerebral organoid, which they're also simultaneously firing. Right, so there are a lot of optical imaging, right? Everybody knows Anna here, right? She used to be working with David Bowles and Sava, right? So so that's a perfect, platforms for us, right, to really go inside experimentally to understanding how the single quantums T2 short, T2 long, right, can potentially alter. Even their partition coefficient will be altered when the activity is different. I think that can guide us to see which part of the biophysical microenvironment, right, will contributing to this unique, T2 star decay-based NARS fMRI signals. But more than that, right, you cannot just ignore the intracellular, extracellular features. You cannot ignore the so-called bound sodium versus unbound sodium, right, which is really contributing to the so-called 3/2 spin multi-quantum studies, right? So luckily, right? So from my German co-workers, right? Many of you, if you know Lothar Schardt, he just retired last year, and but Frank now is a head there. So they published a paper just last year, right? So what they did is nice paper. So basically, what they claiming is, based on the single quantum FID, right, you can mathematically deriving the 3 quantum dynamic features, the 3 quantum signals, right? So they have all these mathematical, why they can prove in the equations, right, based on the, so forgive me, right? So based on the original, FID dynamics versus the Fourier transformed, right? When they are performing certain kind of subtractions, they can prove, right, to coming from single quantum to the triple quantum, right? So from these features, right, what they are showing here is, the single quantum is the blue curves. By perform derivatives, they are able to, the red curves, they can retrieving the 3 quantum features. Okay, so we just simply sending them our organoid T2 weighted data, right? MG sequence, UT sequence, right? It's easy, right? So this data just occurred this morning. I'm so happy about it. Even though I'm not even sure it's right? But I want to give you the insights, right? So it looks like they don't know what's going on, but it looks for me it's very striking, right? So this is a real MR2, which is maybe 5 millimeter, right? So organoid is here, only here, okay? You see there are some darker areas. This just showing the single quantum FID fitting, nothing fancy. When they performing their analysis, right, they were like seeing what's going wrong with your data. You don't see anything. They all looks like unbounded sodium, right? In aggregates, because all the signal is lost, except there are some signal that are showing the 3 quantum features. See there? That's exactly located in the organoid areas. So sorry, right? So this is really very nice, right? When you don't, you still need more communication, but I already see some futures, right? So this means we are able to, by the way, I want to see 3 quantum, because that feature indicating how the sodiums interacting with large proteins more, unbalanced, to exchanging the energy states, right? So maybe that, I don't think it's only intracellular, just let you know, right? Because it's still too small, right? But even the extracellular, right? How exactly, right? These 3 quantum features will be, right? At least they can tell us, you see some signal drop, right? Maybe because during the action potential, right? The 3 quantum indicating the environment has been changed or can contributing to a certain kind of dynamic changes there, right? So that's why, I'm very excited about it. Okay, sorry, I lost the track of the time, but, definitely I want to acknowledge, many people here, particularly, in our lab, right? In the past over than 5 years, not only we have a very good postdocs here, many of them left, but there are new people coming, and also we do, make an effort to train the younger people. I already see a few younger interns and even volunteer students are here, right? So they are either go for college or they are go for MD or PhD programs, and good luck for them. Thank you very much.